The overall goal of this procedure is to reconstitute a typical G-protein coupled receptor into large unilamellar vesicles and measure its ability to carry out ATP-independent translocation of phospholipids across a lipid bilayer using a fluorescence-based assay.
This method can help answer key questions in the intracellular lipid transport field, such as what is the molecular identity of proteins that can scramble or flip lipids, and what is their mechanism?
The main advantage of this technique is that it is very reliable and robust once the reconstitution procedure is established for a given lipid composition and a preferred detergent.
To begin this procedure, use a glass syringe to add 1435 microliters of a 25 mg per milliliter solution of POPC in chloroform to a round bottom flask. Add 160 microliters of a 25 mg per milliliter solution of POPG in chloroform to obtain 52.5 micromole lipids in a 9:1 molar ratio of POPC to POPG. Next, dry the lipids for 30 minutes using a rotary evaporator set to 145 rpm. Once drying is complete, transfer the flask to a vacuum desiccator for 3 hours at room temperature. Then, add 10 milliliters of buffer A to hydrate the dried lipid film. Gently swirl the flask until a homogeneous turbid suspension is formed. Sonicate the suspension at a frequency of 40 kHz in a room temperature water bath for 10 minutes. Then, using an extruder, pass the sample through a membrane with a 400 nanometer pore size 10 times. Follow this with a second cycle of extrusion, passing the sample through a membrane with a 200 nanometer pore size.